Hi, welcome back. We are going to talk about the cerebellum. There you have an image where you see is a dorsal view of the brainstem with the cerebellum of a dog, and you see the cerebellum just there. Cerebellum is a word that comes from the Latin small brain. First, we'll begin by talking about the parts of the cerebellum. Here you have a dorsal view, and the cerebellum can be easily divided in a vermis because it has the shape of a worm. Here is marked in red, and two cerebellar hemispheres in blue. Remember that the cerebellum is located caudal to the cerebral hemispheres. If we make a mid-plane section through the vermis of the cerebellum, we identify that the gray matter is forming the superficial layer, is a cortex, and the white matter is inside. And the white matter remembers the trees, the branches of a tree. So this is why it is called the arbor vitae. And we have different portions. We are going to go from rostral to caudal following this curve. Okay. First, we begin by the lingula. Then comes the lobus centralis. And then the culmen with two portions. Then there is the primary fissure. Primary fissure is a landmark to divide the cerebellum in lobes, as we will explain later. Then we have the declive, the folium, the tuber of vermis, the pyramis, the uvula, and the nodulus. Of course, the cerebellar hemisphere, it also has parts that are named here. Number 8 is the quadrangular lobule. Then we have the simplex lobule and the unciform lobule with two portions, one rostral crus and a caudal crus. The paramedial lobule and the dorsal paraflocculus and the ventral paraflocculus, and the flocculus underneath the ventral paraflocculus. These parts of the cerebellar hemisphere are related to the parts of the vermis of the cerebellum. So the quadrangular lobule is related to the culmen. The simplex lobule is related with the declive. The rostral and caudal crus of the unciform lobule are related to the folium and tuber, respectively. The paramedian lobule is related with the pyramis, as well as the dorsal paraflocculus, also related with the pyramis. The ventral paraflocculus is related with the uvula, and the nodulus to the flocculus. In this, you have a dorsal section of a fixed brain and a dorsal section on MRI. It's a T2 and Hayes sequence. And we identify parts of the vermis. The first is the lingula. And then caudal to it, we have the nodulus and the uvula. Another form or another way of dividing the cerebellum, more clinically oriented, is dividing the cerebellum in lobes. And here, this dotted line marks the difference, or the line marks the boundary between the rostral lobe and the caudal lobe. Here you have the yellow arrow marks the primary fissure. And so all this part of the vermis of the cerebellum that is in red, it forms part of the rostral lobe of the cerebellum. The part of the cerebellar hemisphere that belongs to the rostral lobe is the quadrangular lobule. In blue, we mark the nodulus and the flocculus, and the flocculonodular lobe is another one. And finally, we have the caudal lobe of the cerebellum, here marked in green. We'll focus in the flocculonodular lobe for a better understanding of this small lobe that is so important because it's related to equilibrium. Then we have, on the right, we have a midplane section of the vermis of the cerebellum. As you see, the cerebellum sits on top of the pons and medulla oblongata. Here we identify the choroid plexuses of the fourth ventricle. Remember that the cerebellum, it is not just inside the ventricle. There is a thin lamina here. It's the dorsal and caudal medullary velum. And also here, the choroidal plexus are inside the fourth ventricle. So this part of the vermis sits on top of the tela choroidea of the fourth ventricle. So you see, this mark in blue is the nodulus that corresponds with the flocculus. As you see, the flocculus is underneath the ventral paraflocculus. There are also other structures here, as the trigeminal nerve section, the facial and vestibulocochlear nerves, and the choroidal plexuses of the fourth ventricle, because I have said in other lessons that these choroidal plexuses of the fourth ventricle extend through the lateral openings of the fourth ventricle in the subarachnoid space. More images here, you see in blue the flocculus and the nodulus. And as you see, the flocculus and the nodulus, again in all of them. The flocculus and the nodulus, and the same. And in this video, I'm going to show you the flocculonodular lobe. We have to look under the ventral paraflocculus. This is the nodulus and this is the flocculus. So as you see, this is the vermis and this is the nodulus. This area I'm pointing here. The ventral paraflocculus, the dorsal paraflocculus, and underneath the flocculus, okay? So the cerebellum is connected to the brainstem through the cerebellar peduncles. And we have the rostral cerebellar peduncle here that connects the cerebellum to the mesencephalon. In green, I have marked the middle cerebellar peduncle that connects the cerebellum to the pons. And finally, in blue, the caudal cerebellar peduncle that connects the cerebellum to the medulla oblongata. As the cerebellum keeps ipsilateral connections with musculoskeletal apparatus and vestibular nuclei, and has to inform and also has to receive information from motor systems, pyramidal and extrapyramidal systems, it is contralaterally connected with these motor systems. And all these connections form the cerebellar peduncles. So these cerebellar peduncles are rostral, middle, and caudal. Rostral, middle, and caudal, right? And look, the rostral is formed by afferents and afferences and efferences. Efferences mainly, and afferences from the spinal cord, especially for proprioception. And the efferences, they run up to prosencephalic centers and to the red nucleus of the mesencephalon. Remember, as I have said before, that the efferences cross the midline at the decussation of the cerebellar peduncles. The middle cerebellar peduncle is made just by afferences, fibers that enter from the pons to the cerebellum. They form what is known the cortico-pontine tract, needed for correct movements, right? Because this information is coming from the cortical areas, coming down, forming part of the crus cerebri, and reaching forming the longitudinal fibers of the pons, reaching the pontine nuclei, and then through or by the fibers, the fibers that enter from this pontine nuclei enter through the middle cerebellar peduncle, enter the cerebellum. These are the transverse fibers of the pons. The caudal cerebellar peduncle, sorry about that, it should be caudal, no chidal, is formed by afferences and efferences. Afferences from many areas, olivary nucleus, vestibular receptors, spinal cord, etc. And efferences to most nuclei of the brain stem, ipsi and contralateral. This is very important. So let's look at the cerebellar peduncles. The first is the rostral cerebellar peduncle, also known as brachium conjunctivum. And as you see in this dissection, this is a mid-plane section where you see I have removed the lingula, part of the lingula. You see the mesencephalic aqueduct, the pons, the medulla oblongata, and this is the fourth ventricle. And as you see, these fibers that connect the cerebellum rostrally with the mesencephalon form the brachium conjunctivum or rostral cerebellar peduncle. In a transverse section, you'll see this image with the rostral cerebellar peduncle located medially to the middle cerebellar peduncle. Okay? And in this Balchowski image, you see the rostral cerebellar peduncle and the middle cerebellar peduncle here, and the trigeminal nerve. Okay, again, if you compare it with an MRI, you see, remember that the middle cerebellar peduncle has a medial situation. It is located medially. This is the rostral cerebellar peduncle. The middle cerebellar peduncle, also known as brachium pontis, because it connects the pons with the cerebellum. To be able to identify this in a dissection, I had to remove the dorsal and ventral paraflocculus. That is the mesencephalon with the caudal colliculus, rostral colliculus. There you have the trapezoid body, the pyramids, the trigeminal nerve, facial and vestibulocochlear nerves here, facial and vestibulocochlear. And in a transverse section, the middle cerebellar peduncle or brachium pontis is this one. As you see, is located lateral to the rostral cerebellar peduncle. Is the one here pointed with the white arrow. The caudal cerebellar peduncle is quite big, and in this transverse section, it is easy to identify at the level of the trapezoid body. That is, remember the trapezoid body, these transverse fibers here. This is the facial and vestibulocochlear nerve, right? And this is the lingula of the cerebellum. Vale? The ventral paraflocculus, dorsal paraflocculus, and the flocculus here. On an MRI, it is located in this area here, the caudal cerebellar peduncle. And again, in a dissection, you see, to be able to identify these cerebellar peduncles, I had to cut the brachium pontis. Then you identify the rostral cerebellar peduncle or brachium conjunctivum and the caudal cerebellar peduncle or corpus restiforme and corpus juxta restiforme. The corpus restiforme is the lateral portion for afferent fibers, and the medial portion of the caudal cerebellar peduncle is the corpus juxta restiforme for vestibulocerebellar and cerebellovestibular fibers. Again, on a transverse section, this is at the level of the trapezoid body. Remember, this is the rostral olivary nucleus, or also known as dorsal nucleus of the trapezoid body. Remember, the spinal tract of the trigeminal nerve, the nucleus of the spinal tract of the trigeminal nerve, the rubrospinal tract. These are the fibers from the facial coming from the internal genu of the facial nerve and running ventrolaterally to get out or to leave the brainstem. So, in sagittal sections, we can identify these cerebellar peduncles as well. The brachium conjunctivum or rostral cerebellar peduncle is this one here, is these white fibers. And as you see, there are fibers that are, most of the fibers are leaving the cerebellum. Some of the fibers are entering for unconscious proprioception, coming from the spinal cord, but most of them are leaving and crossing here, ventral to the caudal colliculus of the mesencephalon, crossing the midline in the decussation of the rostral cerebellar peduncles and going up to prosencephalic centers. And in this image here, you see the caudal cerebellar peduncle, right? Or corpus restiforme or justa restiforme, connecting the cerebellum with the medulla oblongata. More things you see here is the accumbens nucleus, the caudate nucleus, the thalamus, parahippocampal gyrus here, parahippocampal gyrus, and then the hippocampal formation with the fornix fibers that leave the hippocampus, the caudate nucleus, fibers of the internal capsule here, okay? So, again, transverse sections, what do we see? We see the fourth ventricle, choroidal plexuses of the fourth ventricle, and we are at the level of the medulla oblongata, the pyramids, right? And then, you know, this is the stria acoustica, the caudal, this area here, that is brownish, is the caudal cerebellar peduncle, the spinal tract of the trigeminal nerve, with the nucleus of the spinal tract of the trigeminal nerve, the rubrospinal tract here, okay? Remember the caudal cerebellar peduncle. Remember this image from other lessons with the dorsal cochlear nucleus and the stria acoustica going on top of the caudal cerebellar peduncle. The stria acoustica is this one here. So on a transverse MRI, this area here should correspond to the caudal cerebellar peduncle. And more ventrally, we should have the spinal tract of the trigeminal nerve with the nucleus of the spinal tract of the trigeminal nerve. And this area in the middle is the raphe. A lot of neurons are located in the raphe as serotoninergic neurons and the pyramids. So the cerebellum is made of gray matter and white matter. The gray matter forms the cortex and cerebellar nuclei, and the white matter forms fibers. So the cerebellar nuclei are three on each side: fastigial, interpositus, and dentate. Here you have them. The fastigial nucleus is the more medially and with a high position. This is why it is called fastigious, because meaning roof. Then we have the interpositus. Here the interpositus. That means in between. In man, it is divided in emboliform nucleus and globosus nucleus. And then we have number 3, that is the dentate nucleus, meaning tooth-like, right? It's the dentate nucleus. So as you see, these nuclei are very close to the ventricle, okay? Remember the development, okay? So, can we identify these nuclei? Well, it's difficult to see them on MRIs. However, we know where they are. This is a mulligan staining, and we see these bluish areas and correspond to the cerebellar nuclei. So we have the fastigious nucleus, and also it has a small elevation here, interpositus and dentate nucleus, or dentatus. And here, the caudal cerebellar peduncle. So on MRI, we should say this nucleus located here. Here we should find the fastigious or fastigial nucleus, the interpositus and dentatus or dentate nucleus. Again, compare. This is a dorsal section of a fixed brain, okay? And we are going to focus here in the cerebellum. Here are the caudal colliculi of the mesencephalon. We also see other things, no? Both thalami, the putamen here, the putamen, the caudate nucleus, the telencephalic septum, right? Okay, and the hippocampus and parahippocampal gyrus here. So we'll focus here, and you see these brownish areas that correspond to cerebellar nuclei, the fastigious and the dentate, right? And also on MRI, we should say that in this area, we should have these cerebellar nuclei. This is a special case because it has a degenerative process that allows us to see these nuclei here that correspond to the fastigial nucleus of both sides, right here. Again, on MRIs, when we see this image on an MRI compared with a dissection, it is from this image here. So we have enlarged this area and we see the rostral cerebellar peduncle. So the rostral cerebellar peduncle should be here. And as we are at the lateral aspect, the dentate nucleus. So we should have the dentate nucleus here. So now I am removing the dorsal and ventral paraflocculus. And we identify the brachium pontis, or middle cerebellar peduncle, with the trigeminal nerve here. And here we have the flocculus. Inside we have the rostral cerebellar peduncle, the caudal colliculus, the rostral colliculus, and middle geniculate body. Again, this is the flocculus. And when we move the flocculus, we identify the stria acoustica going on top of the caudal cerebellar peduncle. This is the stria acoustica, trapezoid body, vestibulocochlear nerve. The stria acoustica is for audition. And the superficial arcuate fibers that we have seen at the brainstem lesson. The primary fissure that separates the rostral lobe from the caudal lobe. This is the primary fissure. Okay, and the brachium pontis. Okay, cerebellar circuitry. This is a drawing of the layers of the cerebellum. Okay, and there are two types of fibers that enter the cerebellum. One are the mossy fibers that are glutamate, so they're the excitatory. Okay, these mossy fibers, some of them project to the cerebellar nuclei, and some axons go up to act on granular cells. These granular cells project their axon to the molecular, to the upper layer, okay, to the upper layer, that is the molecular layer. And they are excitatory over two types of cells. One are the Purkinje cells, and the other are the basket cells that are GABAergic. These basket cells inhibit the Purkinje cells, so it's a regulation process of the activity of the Purkinje cells. These Purkinje cells project their axons to the Golgi cells at the granular cell layer, and the Golgi cells are GABAergic, are inhibitory over the granular cell layer, over the granular cells in the granular cell layer. And the axons also of the Purkinje cells, they go down and they may do two things. One is end in cerebellar nuclei. Another is some of them, they can project directly to ipso and contralateral vestibular nuclei. Other types of fibers are the climbing fibers. These climbing fibers, they come from the olivary nucleus that is in the medulla oblongata. Again, these climbing fibers, they project to the cerebellar nuclei, but they also go up. Some of them project collaterals to the granular cell layer, but they go straight to the Purkinje cells. And they are excitatory. They are the neurotransmitter is the aspartate. And it seems that these climbing fibers are involved in learning processes. The difference between the MOSI and the climbing fibers is quite obvious. In the case of the MOSI fibers, each Purkinje cell receives input from approximately 200,000 granular cells, and each granular cell receives inputs from many MOSI fibers. However, in the case of the climbing fibers, that they are coming from the olivary nucleus, each climbing fiber contacts with 1 up to 10 Purkinje cells. It's more precise. So, now we are going to talk about the afferents and efferents from the cerebellum. And in order to do that, we have to remember that there are several types of cerebellar divisions, and this is going to be very important for understanding the afferents and efferents. First, remember there is a longitudinal division. I said that the cerebellum is divided in a vermis and a cerebellar hemisphere, and the part of the cerebellar hemisphere that is close to the vermis is the paravermis. Another one was the lobular division with a rostral lobe, a caudal lobe, and a flocculonodular lobe. However, phylogenetically, we can say that there is 3 types of the cerebellum. One is the paleocerebellum, that is formed by the lingula, central culmen, and quadrangular lobule, and the dorsal paraflocculus, ventral paraflocculus, the pyramids, and the paramedian lobule. There is an archicerebellum, also known as vestibulocerebellum, and is formed by the archicerebellum. And finally, there is a neocerebellum or cerebrocerebellum that is formed by the rest of the cerebellum. Okay. So, for the afferents, it's better to consider a phylogenetical division. So, in the case of the spinocerebellum or paleocerebellum, it receives MOSI fibers from the spinocerebellar tracts and climbing fibers from brainstem nuclei that are involved in keeping the muscle tone. So it's very simple, right? Just to keep steady and keep fight against gravity. It's important, and to keep the tone of the muscles, and to receive most of the information from the spinal cord. The neocerebellum or cerebrocerebellum receives just MOSI fibers, no climbing fibers, and from corticopontine tracts, and is involved in cognitive areas, visual, motor, somatosensory, etc. And finally, the archicerebellum or vestibulocerebellum receives MOSI fibers from vestibular tracts and climbing fibers from optokinetic centers in the brainstem. These are related to the eye movement to compensate the head movement. In particular, I have pointed out to the pyramidal pontine reticular formation for the saccadic generating center and the rostral colliculi for visual reflex. So the vestibular nuclei project MOSI fibers, okay, to the cerebellum. Okay. Now, for the efferents, we should consider the longitudinal division. What I mean? The flocculonodular lobe projects through the caudal or caudal cerebellar peduncle, and they are ipsilateral to vestibular nuclei directly. So they participate in equilibrium. However, the vestibular nuclei, they also receive fibers from the cerebellar vermis that they do not go to the fastigious, and what they do is they go straight to the vestibular nuclei, and they go through the caudal cerebellar peduncle. Okay, so lesions in these areas may end up having vestibular signs, broad-based stance, and sway from side to side at gait. The cerebellar vermis, what do we have under the cerebellar vermis? The fastigious, the fastigial nucleus. Right? The fastigial fibers from the fastigial nucleus live through the caudal cerebellar peduncle, and they go ipso and contralateral. And they end in descending motor systems and centers that are involved in keeping a stance and muscle tone. So they are involved in maintaining muscle tone, posture, and eye movements. So a lesion in these areas may result in an increased muscle tone, okay? Extended rigidity and opistotonus and titubation, okay? This rhythmic oscillation of the head of the trunk itself, or the trunk itself, sorry. Paravermis, which is the nucleus that is lateral to the fastigial, is the interpositus. And lateral to the interpositus, the dentate nucleus. So the paravermis projects to the interpositus and the rest of the cerebral hemispheres to the dentate nucleus. Both dentate and interpositus are projecting through the rostral cerebellar peduncle or brachium conjunctivum. They cross the midline at the decussation of the rostral cerebellar peduncles, and they go to the prosencephalic centers, pyramidal and extrapyramidal centers, and also to other areas, but also involved in the controlling the voluntary movement. So a lesion here may end up, and it is important for synchronized coordination of the movements. Okay. So a lesion here, we may end up having intention tremor, okay, this dyskinetic disorder, okay, and cerebellar hypermetria, an exaggerated movement of flexion and return to ground with an excessive extension of the limbs. Okay. So, summarizing, we say that, look, these deep cerebellar nuclei receive afferences from climbing fibers, okay, coming from the olivary nucleus, mossy fibers from the rest of the fibers. And also, they receive information, inhibitory information from Purkinje cells, because the Purkinje cells, they also receive climbing fibers and mossy fibers information. So that these deep or cerebellar nuclei are receiving excitatory information from mossy and climbing fibers and inhibitory information from Purkinje cells, and they act excitatory. So look, so they need this Purkinje cell information to inhibit and to control the excitation or the activation or the function of these cerebellar nuclei cells. So fastigial, interpositus, and dentate, right? And these ones are projected to prosencephalic and pyramidal and extrapyramidal motor centers, and the fastigial to brainstem nuclei to keep this tone of the muscles. So remember, the cerebellar nuclei project excitatory activity. However, they receive inhibitory activity from the Purkinje cells. This is a drawing that may summarize their relationships. Okay, fastigial, interpositus, and dentate nucleus. Look, the middle cerebellar peduncle here receives, it is formed by fibers that are coming from the pontine nuclei. Remember that these pontine nuclei receive through the corticopontine tract fibers from the motor, sensory, and visual cortex. All these fibers, they run up to the pontine nuclei, and they are projected through the fibers from the pontine nuclei to transverse fibers. They form the middle cerebellar peduncle and they enter in the cerebellum. The caudal cerebellar peduncle, okay, it's receiving fibers from vestibular nuclei, right? Of course, they also receive other things from the spinal cord, proprioceptive centers, etc. Right? However, through the caudal cerebellar peduncle, fibers from the fastigial nucleus cross the midline, and they end up in a lot of nuclei of the brain stem. So as you see, the fastigial nucleus is projecting to many nuclei of the brain stem, even the red nucleus, the thalamus, and ending in the cerebral cortex. Okay. However, the interpositus and dentate, they go through the rostral cerebellar peduncle, they cross the midline, and they end up in the red nucleus, thalamus, and the thalamus is projected to other areas of the cerebral cortex. So this interpositus and dentate, mostly the fibers that leave these nuclei, they mostly go up to the pyramidal, extrapyramidal, and other pyramidal centers. Okay. This is important. This is cerebellar somatotopy. This was taken from this paper, but it's interesting. You will see how later on. The thing is to map the cerebellum as the cerebrum has been mapped, and many drawings have been done in the cerebellum. However, multiple representations of the same body parts are in different locations, and this is a big mess. Okay? But summarizing and simplifying this, we found that, as you see, we have here in blue the rostral loop. Here the rostral loop is here. This is from human and this is from a dog, right? So the vermis of the rostral loop seems to be involved in keeping the muscle tone or regulating the muscle tone of the head, the shoulder, and the upper trunk. And and look at the limbs. Of course, there are feet as well, but there are hands, the bigger hands. So the cerebellar hemisphere, the cerebellar hemisphere from the rostral lobe of the cerebellum, okay, coordinates forelimb, face, and mouth. Okay, forelimbs, maybe forelimbs, right? This here, okay. So, and the vermis of the caudal loop is involved in lower trunk and pelvis, and the rest of the cerebellar hemisphere from the caudal lobe is involved in the hind limbs. Okay, so this is important because somatotopy versus dysfunction, lesions that affect the rostral lobe, we should think that the signs should be affecting the head, neck, and control of the forelimbs. However, if the lesion affects the caudal lobe, we should be expecting pelvis, lower trunk, and hind limbs disorders. And when the lesion affects the flocculonodular lobe, we should expect disturbances in equilibrium dysfunctions. Okay? So, here I want to point out a special circumstance or situation. Look, this is to represent the vestibular root from the vestibulocochlear nerve projects vestibular information to the vestibular nuclei, right? Rostral, caudal, and medial vestibular nuclei. These rostral, medial, and caudal are projecting information through the vestibulo-thalamic connections to be conscious, okay? And seems to be bilateral. Also, there are fibers that bypass these vestibular nuclei and end in the flocculonodular lobe, in the fastigious, and in the rostral lobe of the cerebellum. Okay? Here here I represent the fastigious projecting to nuclei of the brainstem, ipsi and contralateral. However, look at that. In green, the flocculonodular lobe projects inhibitory GABAergic neurons to the rostral, caudal, and medial vestibular nuclei. These nuclei project through the medial vestibulospinal tract. Okay? And these are important. Okay. The lateral vestibular nucleus receives inhibitory GABAergic neurons from the lateral strip of the rostral lobe. Okay? And this lateral vestibular nucleus projects through the lateral vestibulospinal tract. Okay? And this is responsible for the increasing of the extensor tone towards the lateral side or the ipsilateral side for anti-gravitatory purposes. I mean, if you fall to the left, you have to increase muscle tone on the left. Look, turning the head to the left causes a shift in the distribution of the weight, okay? And if you turn the head to the left, you increase you shift the weight to the left. So you have to increase the tone on the left. And this increase in the anti-gravitatory muscles to the left, and there is a tilt movement to the right. And this tilt movement to the right, the responsible of this tilt movement to the contralateral side is responsible is the medial vestibulospinal tract, this rostral, caudal, and medial vestibular nuclei. These are the responsible for the turning of the head towards the contralateral side. Okay, so what's going to happen? If you have a lesion that affects the flocculonodular lobe, then this inhibitory activity of the flocculonodular lobe over the rostral, caudal, and medial vestibular nuclei that project through the medial vestibulospinal tract are exaggerated because there is not inhibitory activity. So in this case, the animal is going to show a head tilt contralateral to the lesion. Okay, so we are having a paradoxical vestibular syndrome. And if the lesion is affecting the caudal cerebellar peduncle, then it affects the fibers that are coming from the lateral strip of the rostral lobe of the cerebellum that are inhibitory over the lateral vestibular nucleus that projects its action through the lateral vestibulospinal tract to increase the muscle tone ipsilaterally. Okay, so we end up having what? That. So what do we have? We have, in this case, vestibular signs to the right and increasing muscle tone to the left. And this was caused, in this case, by a lesion affecting the caudal cerebellar peduncle on the left. Okay? So again, we may found that. What do we have here? Opisthotonous, increase in muscle tone on the forelimbs, and flexed hind limbs and flexed hip. Okay, these are the cerebellar signs. Again, we have opisthotonous, increased muscle tone in the forelimbs, flexed hips, and in this case, it was a compression of the rostral lobe of the cerebellum by a cyst. Right? Compare the cerebellum of a sheep with the cerebellum of a dog. This is a mid-plane section through the vermis. As you see, this is the rostral lobe, and here the rostral lobe seems to be bigger than in the dog. However, the caudal lobe seems to be smaller in the sheep than in the dog. Again, another case. Okay, opisthotonous, increased muscle tone in the forelimbs, increased muscle tone in the hind limbs, but flexed hip. In this case, was an abiotrophy of the cerebellum, and very few Purkinje cells were able to work. So again, if we go back, we remember that lesions that affect the rostral lobe, okay, are going to show signs in the head, neck, and upper limbs. And if the lesion extends to the caudal lobe, appear signs in the pelvis, lower trunk, and lower limbs. Okay. Okay, for a better understanding of this, the cerebellar rigidity, remember that there are motor nuclei in the brain stem. Okay. So we have the red nucleus, okay, with the rubrospinal tract. The rubrospinal tract that is facilitatory. We have vestibular nuclei with the vestibulospinal tract that is facilitatory over extensors. We have the pontine reticulospinal tract that is excitatory over extensor muscles. We have here the medullary reticulospinal tract, medullary reticular formation, that is inhibitory over extensor muscles. And this, in this case, to work, it has to be activated by upper centers. And vestibular nuclei and pontine nuclei that work in synergism, okay? They are excitatory over extensor muscles, right? And they don't need to be activated by higher centers. Remember that all these neurons are ending in the alpha and gamma motor neurons that are the lower motor neuron system. The gamma motor neurons innervate the intrafusal fibers that they send signals to the central nervous system in order to proprioception and to activate the alpha and gamma motor neurons. Okay. But something, the cerebellate rigidity shows opistotonous extension of forelimbs and or hind limbs, and in red, flexed hip. This is why is a flexed hip. Summarizing, remember, the red nucleus, they all act on interneurons, and they act on alpha and gamma motor neurons, and the red nucleus facilitates flexors and inhibits extensors. The lateral vestibular nucleus, they mostly act on alpha motor neurons and they facilitate extensors. The pontine reticular formation works synergistically with this one, with the vestibular nucleus, and facilitate extensors, but in this case, acts mainly on gamma motor neurons. Medullary reticular formation, it's inhibitory over extensors, and they act mainly on gamma motor neurons. And finally, the corticospinal tract that acts on alpha and gamma motor neurons and facilitates distal flexors and inhibit distal extensors. They act mainly on distal muscles of the limbs. Then they act for fine movements. So let's go and remember the alpha and gamma coactivation. Remember that the amber motor neuron acts on alpha and gamma motor neurons. Remember that the gamma acts on the intrafusal fibers, and these intrafusal fibers is sending signals to the alpha motor neuron, and they, in turn, these act on extrafusal fibers. When we have an increase in muscle tone, if we cut the dorsal root, and the gamma, the rigidity disappears, so we say it's a gamma rigidity. That it means that the gamma motor neurons, they were the most important here. Okay, but however, if we cut the dorsal root and the rigidity does not disappear, we say that the most important is the alpha motor neurons. So we have an alpha rigidity. And for here, what do we see in the case of the decerebellate rigidity? That we end up having an alpha rigidity. Okay, these vestibulospinal tracts act on neurons, on motor neuron, and on alpha, mainly in alpha motor neurons. Okay, so we have an alpha rigidity. Then this is caused by not by the cerebellum, that does not work, that does not inhibit these vestibular cells, okay, these vestibular neurons that project to the alpha motor neurons. However, if the lesion affects the rostral lobe of the cerebellum, we should find signs on the neck and on the rostral limbs. And if the lesion extends and affects the caudal lobe of the cerebellum, the signs may be extended to the hind limbs, so extension of the hind limbs. So again here, so we have this area is controlled by the rostral lobe of the cerebellum, and this area is controlled by the caudal lobe of the cerebellum. Okay, and here we have two cases of decerebellate rigidity. In the first case, opistotonos, as well as in the second, extension of the forelimbs, as well as in the second, and here in the cat, a flexed hind limbs, but in here is we have an extended hind limbs. But what do they have in common? A flexed hip. We understand the hind limbs depending on the parts of the cerebellum that are affected. But what about the flexed limb? That is common in this situation. So we go back, this is the column, and underneath we have hypaxial muscles. The function of these hypaxial muscles is to fight against gravity. So gravity flexes the column, so the hypaxial muscles are extended, and in a reflex, myotatic reflex, they are contracted. They contracted and they keep the column in place. One of these hypaxial muscles is the iliopsoas muscle, okay? That is the responsible for the flexion of the hip. What is going to happen? In the cerebellar regions, the cerebellum does not control this reflex of the hypaxial muscle. So we have an increased muscle tone of the hypaxial muscles. And one of them is the iliopsoas muscle. So we end up having a flexed hip. And I think this is why most of the animals that have a cerebellar lesion, okay, they tend to flex the hip and fell down. Yeah, right. Okay. Thank you very much.